It's an enormous pleasure today to have our guest speaker, Morten Meldal, who many of you will know as a Danish chemist, and he won the Nobel Prize for click chemistry in 2022. He's a professor of chemistry at the University of Copenhagen and also the Carlsberg Laboratory, which is, again, a very famous chemistry laboratory in Denmark. His work has mainly been in organic chemistry, particularly in peptide synthesis. And as I said, he won the Nobel Prize in 2022 as one of the pioneers of click chemistry. However, today he's going to put that in perspective of you cannot do research without the use of infrastructures. So everybody on the call here, we're all interested in research infrastructures. And Anita Hewitt and myself were very fortunate to be at the Danish presidency meeting on research infrastructures last year, where Morten gave a very inspiring speech. I think it was true to say that Anita said it was one of the things where she agreed with nearly everything that was said in the presentation. And I hope we're going to hear about that today, and the use of infrastructures and the need for infrastructures, particularly perhaps some of the smaller scale infrastructures, not the giant CERNs, in the development of research, which can lead to pioneering research, which of course can also lead to the ultimate goal of applications and of course maybe winning a Nobel Prize. So Morten, we're delighted to have you today. Thank you for giving up your time, and we're very much looking forward to your talk.
Well, Nigel, thank you very much for the invitation to actually come here today. I have called this infrastructure and the key performers of our economy. And as I will outline, it's because I have a strong belief that education and research are the key to a successful society in the long timeline. So chemistry is everything, and I mean that very literally. In the sense that everything from working up rare earth minerals to working with wood or even medicine, our health, our emotions, our food, everything is chemistry, molecules that come together, and when they come together they recognize each other, they undergo transformations with enzymes. We have molecular signaling that leads to our function. Essentially, so if we feel pain, if we feel cold, if we feel warm, and so on, all of that is molecular signaling in our bodies. And then you have molecular transport getting into the cells, getting to the targets, and then you have molecular replication, which of course is an essential part of life and evolution. So if chemistry is really everything, I think it's extremely important that we do a strong investment in this area, in particular in the education. So we should have the young people from the very early stage of their schools learning about this chemistry, not as formulas from the beginning, but as a visualization of the chemistry world, a 3D visualization. And we should also do research without constraints, because the chemistry of both life and also chemistry around us is too complex for us to actually understand it. And mostly the breakthroughs comes as a surprise. So it's serendipity that leads to the new great discoveries that drive our society forward. And for that reason, we also should have funding on a European scale. We should have funding by performance. So if you did very well the last 3 years, you can continue doing very well for the next 3 years. I think that's a very simple model. There are 3 products from universities, the bachelor's, the master's, and the PhD candidates. Then there is the research publications and the innovation. And these 3 products is what we should really focus on in the build-up of our research and university facilities, because they are the ones that are driving the economy in the society. So there's another thing, and that is that the world is an increasingly dangerous place. Why is it becoming increasingly dangerous? It's partly because of our overconsumption and global warming as a result of that. But it's also because when there is a threat, we as human beings try to avoid that problem, and often by backing up behind a strong person. This is a real problem, and it's only avoided by education. So we have in the world today the autocratic trend, which is leading to a world that is essentially ruled by narcissists. They involve themselves, so we have an example right now, in senseless wars. We get a community crisis because both because of our leading into the world of entertainment, but also because of the large-scale harm that happens in the society. There's no empathy left, and empathy is what is driving our survival, what is driving our success as herd beings. And the global warming consequences are much more severe than the consequences that we have of political issues at the moment. And this leads to ice melting affecting a very large fraction of the population in the world. Plastics everywhere, microplastics, new pandemics, flooding, fires, deserts that expand because we have a hotter atmosphere. So water goes up, goes further out, and comes down in large amounts. We have flooding because of that in places we didn't have flooding before. We are losing diversity, and we have resource depletion. So the only way that we can come around this is education and research. So new paradigms of how we can actually operate in society, and that's education, education, education. So there are three types of infrastructure here illustrated with ESS. The international and national large-scale facilities. Then there is intermediate-scale university research facilities such as NMR and mass spectrometry facilities. Then there's a large amount of distributed small-scale infrastructure that is really, really important for research. So they are not only important for research, they're important for fundamental research, but also for the education itself. If we don't have infrastructure and preferably walk-up instruments for the students, they don't really learn anything. We need to have applied research infrastructure as well. So to transform and translate the fundamental research into innovation and translation into industries. So the focus in our society should be, if we want to solve the problems that are facing us, STEM, and particularly important is chemistry and chemical biology, to solve many of the problems that are associated with us getting older and older and having less and less young kids. So all sorts of societal diseases and so on. But also because STEM is actually what drives the entire economy in Denmark, in EU, in China, and US. The large-scale facilities are doing exciting research, and it's highly valued by also the donation of grants that can allow these large-scale facilities to operate. The intermediate facilities are also considered important. They're crucial for research. We cannot have publications without NMR, MS, and so on. And then there's a distributed small-scale instrumentation, which is really, really essential for research and education. And that part has a real problem. So here's a large-scale ESS, and we have another example here in Denmark, which is a quantum computing center. We have DTU, NMR facility looking like that. And then we have what we actually use every day. This is a lot of instrumentation, and it really is quite expensive to run, to acquire, to run, and to maintain. So This is most important actually for most of the research. 90% of the research is carried out in this part of the infrastructure. It's for acquisition of data that are in every single paper that is published. It's for education of our students in how to operate within the STEM area. It's acquisition of these instruments is usually by VIPs in the facilities or in the institutes. And they have to maintain themselves, these instruments, and that's like 5 to 10% of the purchase annually. And in Denmark, the downtime of instruments like these is really, really significant, and that is because we don't have any core infrastructure facilities. So Denmark has a lot of companies that are directly related to our education of chemists. So 100 smaller chemistry-based companies plus these large one, Novo Nordisk, which is, as you can see here, responsible in already 2022. They are now worth more than the collected Danish BNP in 2022. You can see here how it increases with time. So chemistry and chemical biology education drives the Danish economy and growth. And education has the highest return on investment. I'll come back to that later on. In other countries, you have government-funded infrastructure hubs. And China really has a lead today in research. If we go into the journals, you will see most of the really important research results are actually achieved in China, not in the US, as we have expected for many years US to be leading in that area. I think today we can fairly easily say that China is in the lead. That doesn't mean that China is in the lead financially, economically in the world, but it means that China is doing the fundamental research steps today that will lead to the development of tomorrow. Then we have US and EU with all their dysfunctional infrastructure problems. You know that the NIH funding has been cut. There's not a lot of political will to fund research infrastructure in the US and EU. I don't know about all the countries in EU, but most of the countries have the same system as Denmark, where the EIPs get the small-scale instrumentations by themselves, and they have to maintain that. India is a runner-up, together with Saudi, with the infrastructure hubs functioning extremely well in these two countries, and we can see more and more important research results from there. So the question is, do we consider research infrastructure appropriately? We need equipment for the laboratories, of course, and that is true infrastructure programs from foundations and national and EU funding bodies, and that's fine when we get that as individual VIPs. And then it is essential for performing our daily work and research and studies in the laboratory, but that doesn't really work if the instruments don't work. So the problem is maintenance. How is this maintenance covered? In Denmark, it's from the VIP accounts. So if you don't have funding, suddenly your instruments goes down. Everybody in the institute that use that instrument have no access to the data that they can produce anymore. It's also adding to the running cost in research programs, and their running costs are downgraded with time. It's really difficult to get running costs today because we have to cover all sorts of administrative costs in the universities. Unnecessary in my opinion, actually. And we have no maintenance expertise. We have no insurance for the instruments. We are self-insured. And there is exorbitant repair costs from the suppliers. And the result is very expensive downtime, waste of workforce, waste of critical education time, and reduced quality of the research. So another thing is that the student really needs hands-on which means that the maintenance is increasing. If we need students without experience to have hands-on on some of the instruments, that actually is difficult to do without any maintenance. They have to know their chemistry and biology, they have to make the research and innovation, and they have to get disruptive ideas. They only do that when they are actually doing the experiments. So in order to get into the industrial implementation, you also know how to do that actually, from project to factura, as we said in Denmark a couple of years ago. I have sort of spiced this up a little bit with a beautiful holiday that I had in 2022 in Stockholm. For 1 week we were invited to by the Nobel Prize committee to come to Stockholm and spend time together with other Nobel laureates. And the first day you deliver an important thing. This is a bit of infrastructure that is now in the Nobel Museum. You deliver something that was very important for you, and this is the key to combinatorial chemistry in our laboratory, a multi-well synthesizer that was used for generating many compounds. And you also get to sign a chair in the museum. So this is my signature together with Emmanuel Carpentier, very proud of that. But the inspiration has to start early, and we have to actually inspire the young people, the very young people, because when you are a kid, when you are very young, you have this enormous ability to store images. When we are born, we have no language, we only have our vision to teach us about the complexity and the surroundings that we are entering into. So we built, by imaging, we built a mirror of the outside world, and this mental mirror we are using to navigate the world. We can utilize that to increase the understanding of a three-dimensional space by animation, diving into the chemistry that is in control in this beautiful picture here, diving into the chemistry of life, diving into the chemistry, inorganic chemistry and so on, in 3 dimensions, and looking inside in 3 dimensions. I think this would actually teach the children and inspire the children to learn much more about this reality. Real inspiration comes also from experience, and here is my experience at 15, 16 years. I went down to a shop south of Copenhagen and bought all sorts of explosives, went back with a train, 15 kilos of explosives in a train, to my parents' basement, and I did a lot of successful experiments in the basement until one day I had a huge explosion in the lawn of my parents' house. And then, of course, it was finished with doing experiments in the basement. But it was a lot of fun, and it inspired me to study chemistry more seriously. But the road to scientific success is not straight. You really have to get your fingers wet in order to achieve success. Science always fails initially. You cannot do a design behind an office desk and hope that it will work. So you have to fail and rise again, do it again and again. You cannot really plan, and you have to look for opportunity so that you can use your time optimally to get new results. And you have to find out what you find is interesting, get out of comfort zones, change fields and bring your experience with you, and study all your life. So this is what I think is very important for the young people today, because we are facing so many problems. But the factual knowledge is not the most important thing here. What is more important is that we start to teach also more soft skills like your intuition, follow it, learn how it works and follow it, your imagination. Are you able to actually picture things in your mind? This is something you can train, and I think it's a very important skill to have. Association of very different things in your brain. Being able to observe something that you don't expect That's really hard. In science, that's really, really hard to see the unexpected. And being able to combine these observations with the knowledge you already have, to see if there's any creative results of that process, and have the courage to follow that, even your peers are telling you this is no good, you should still follow it, and have perseverance with your follow-up there. So I would like to take an example of how we use infrastructure in one of our projects. So this here is the TAT peptide, a peptide in HIV virus that the HIV used to penetrate into the cell interior. This is the structure if we use a one-letter amino acid code for that. And this peptide is actually quite toxic because of all these R and K, which are basic residues, basic amino acids, and there's a lot of them in this peptide. So it actually is quite, unhealthy. So we thought maybe we can make something that is better. And we made a small peptide like this by combinatorial methods. We made a whole lot of them, and then we tested out and found some that could bring cargo into cells the same way as HIV molecule can. So we started out by synthesizing this part of the molecule here, the building blocks, and we started with hydroxyproline, a natural amino acid, converted that into ether derivatized, so this big bulb is this one sitting here, for alkylation of the oxygen in hydroxyproline. We also transformed this into amines by azide substitution and reduction. Then we could add on the lipophilic chain here, and so the hydrophobic chain, the fat-like chain here, and then we could protect the building block for synthesis by the FMOC solid phase synthesis. So now we had the building blocks, and in this process we are using, of course, a synthesis facility with all that has. We're also using liquid chromatography, high-resolution mass spectrometry. We're using high-resolution NMR spectroscopy, and we are using preparative purification, HPLC. So this is all instruments that are required to make this compound and that compound. And without them, we couldn't do it. So the next step was to make a variety of different compounds like this using the building block. And here are the building blocks that we used, and these are the peptides that we made. And you can see here that these are cyclic peptides with a click reaction. This is the result of a click reaction in the middle of the peptide to link them all together. And here we are utilizing the ease of doing clicking between alkynes and azides, and we can do this on the solid support in connection with the peptide chemistry. So we synthesized 16 compounds, and we then tested those out on cells to see if they go into the interior. For this synthesis, we needed polymer synthesis equipment, so special equipment for making polymers that are beaded, solid phase peptide synthesizers, LC high-resolution mass spectrometry to confirm the structure, and preparative HPLC to purify them. Without these instruments working, any of these instruments not working would put a complete hold to this project. So here are examples of an LCMS of one of our compounds that we synthesized, quite pure. And this is our reference, a TOTA peptide with a TAT type of compound sitting here. And you can see the result is very high purity, and this is only possible when you have purification equipment. So then we studied these compounds in cells, and there we're using a GMO facility must work with lab benches, and we have plate readers, we have cell incubators, we have all sorts of things in the cell laboratory, but all small-scale equipment. And you can see here we are testing the compounds in cells, and only one of them, this one down here, shows a very good import. I have taken it up here, and now we are comparing with the natural TAT peptide, which also imports to some extent, but you can see our compound that we have generated here is much more efficient than the TAT peptide. Here we are also using very expensive but confocal high-resolution microscopy imaging, and we have to do collaboration. We don't have an infrastructure hub to do that, so we have to do collaborative work in order to get our confocal imaging. And with this we could demonstrate that we have one peptide which is particularly good to get into the cells, and it's about 40% better than the TAT peptide, or even more. So that was a very good result. We could also characterize how it works. We could see that it's energy dependent, so it's an active transport into the cells. We could see that the optimal concentration inside the cells is for 12 hours. And we could see the dependence on concentration in solution to the import into the cells. So these all require a lot of imaging, a lot of cell sorting, and incubators for the cells and so on. Then we needed to test here what is actually the mechanism of this import. And here are some inhibitors that you can use for inhibiting different pathways of the import. And you can see the dinosaur inhibitor here is very active, showing us that this is a dynamine-dependent endocytotic pathway. In order to demonstrate that, we needed a mammalian cell facility, plate readers, cell sorting again, and confocal high-resolution microscopy. And we also studied a variety of different cell lines here in this study. So we wanted to see, can we really import big things with this compound? So we synthesized a clicking compound here, and you can see now we have a lot of atoms in our molecule, so it's getting more and more difficult to completely characterize these compounds. And here we are using protein expression to make a green fluorescent protein with a click residue inserted into it, and we can click it onto our cell-penetrating peptide, which has an alkyne here at the N-terminal. So we click them together, and this is shown down here, CPP18 with the clicking. And when we just mix CPP18 with our green fluorescent protein, nothing comes into the cell. Also, if we just treat it without the CPP, nothing comes into the cell. So the fluorescent protein is not transporting itself. But when we have these two connected, you can see very, very efficient import into two different cell lines here. And for this study, of course, we need protein expression facility. We needed a CPG, it should be CPG protein purification. We need multi-tough MS, and we need high-resolution microscopy. So you can see we need really important infrastructure, and any one of these instruments not working is completely delaying our publication, which came out recently in Angewandte Chemie. In addition to all the laboratory equipment, we also did a lot of modeling in this project, and he has shown some of the mechanism for the import. So we also need high-end molecular modeling here. This is back in Stockholm. This is the day when you give your lecture. This is the day where you get the medal. So you get the medal from the king, and you go back to your seat. On the way back to your seat with your medal it is taken away from you. And then you get it on the last day. And the reason for that is that many of us are much older than when we got the ideas that led to the Nobel Prize. Some of us has lost it in the meantime to Alzheimer or other neurodegenerative diseases. And now you get the medal on the last day and you have to bring it home yourself. So as I said initially, molecular recognition is a very important process. It is involved in every single thing that happens in biology. Normally the master of molecular recognition is the antibody in our biology. Antibodies can actually be made to recognize particular foreign organisms, and that's how we get an immune response. But we thought maybe we can do something much better with small molecules because they are, available synthetically in this scale, in contrast to the antibodies, and also they could be used for combinatorial chemistry. So here we have a molecule which is structurally maintained by the blue residues and green residues, and the red residues can be modified at will to fit to a protein surface. And we did that here on the top and the bottom of a protein called interleukin 6. Interleukin 6 is a immune protein that's very important for a variety of different reasons. And we looked at the recognition here. We mixed this one with a bead, where we have one of the molecules attached to the bead, and the other molecules is attached to a fluorophore. When we mix the two, nothing happens between them. So we have an uncolored bead. But if we add the green and orange protein here, even at very low concentration, 20 nanomolar, you can see the green and orange protein is recruited by the bead, and then the protein that is bind the fluorescent probe from the solution, and we get this fluorescent bead. So we can demonstrate here that we can make molecular recognition using the click chemistry. We can make molecular recognition, and we can make it twice just by simulation in the computer, synthesis, and testing in solution. So what can we use that for? Well, we have some very serious societal problems associated with neurodegenerative diseases, as I just talked about. Here is the example of an Alzheimer plaque in the brain. And Grimm at Allied, they isolated these fibrils from the plaques in the brain and got a crystal structure out of it. So this is an electron microscopy structure. So we need an electron microscope to get that. And we can then split that in two and look at the cross sections. Here is one of our beta bodies sitting on the growth phase of the fibril. But in fact, we need one on each end of the fibril in order to block the growth of the fibrils, because we want to bring these fibrils into solution and remove them if we can during a deep sleep at night. If we use this molecule fluoresces when it comes in contact with this fibril, this beta sheet fibril, and that's what you see here, a fibrillation curve, another fibrillation curve here. And then when we add our molecules, like these two molecules, we see a little bit of fibrillation and then it dies off and goes away. We were not completely happy with this. Oh yeah, we also tried to defibrillate already formed fibrils, and that worked. So we could sort of solubilize already formed fibrils, which is of course the target of this. But we were not totally happy, so Jan Sang, he made a building block for solid phase synthesis which has more hydroxyl groups. And we want these to stick out into the solvent and bring water with it into the recognition process. The nice thing is that this has the same configuration as the structural residues that we are already using in our beta bodies. And when we incorporate them in these beta hairpins, so these are the GAs, you can see 3 here and 2 here. When we incorporate them, we get a very good solubility of the compound, And here you see the recognition with compound 3, 4, and 5. And 3 and 5 were really good in this assay that we have. You can see here the fibrillation curve without addition of anything, and then different additions. And when we add 3 and 5, we get absolutely no fibrillation in the assays. So that was very cool. We have now a molecule that can prevent fibrillation in the brain. And the way it works is by having this water shell. The water shell sort of makes part of the structure. So the structure becomes like one big thing that interacts with the fibrils. So we have this water and the molecule with recognition, and that comes together with the fibrils and dissolve them. In this program, we also looked at the possibility of actually going in and cleaving these peptides that form the fibrils. If we could cut them in two, maybe they would not fibrillate, and maybe we could get rid of them. So Martin Wolfram made what is called fluorescence quench substrates that are part of the fibril peptides. And he also made some catalysts, metal binding catalysts. Here is a carbene that binds to a transition metal, and that is interacting with our substrate, and the substrate is then supposed to cleave here in the middle. And that worked extremely well. We could use all sorts of metals, and we got cleavage both with that and with this one over here. But Martin got suspicious, and he took out the catalyst completely, and it still worked. So we have activity, cleavage of this substrate, even without organosign present. So this is where you don't throw your experiment away, but you actually go in and analyze all the little details and what is happening, and you need the infrastructure in order to do that. So we synthesized all the different fragments that we were observing in solution in order to study this in detail. And what we found is that this is not a very rapid reaction. In the beginning, nothing happens, but then after 11 days, something suddenly happens, and you get these new fragments. And it's the Alzheimer peptide themselves that is doing that. So we discovered an autocatalytic reaction with the Alzheimer peptides. They catalyze their own destruction. And here we see small fragments that we can actually observe in the mass spectrometer when this is happening. Moreover, we can take this active mixture here, just 1 microliter, and add it to a well with a longer peptide from the Alzheimer, like here. And you see after a lag period, we have complete destruction of these longer peptides. We could even take the full-length Alzheimer peptide. When we do a little bit of seeding, in 12 days we have complete destruction of the Alzheimer peptide. And even the 1 to 42, which has not previously been reported to do anything like that, and it didn't do it for 3 months, but Lea Kaufmann was persistent, so she came back after 3.5 months, and suddenly we saw this result, complete degradation of the Alzheimer peptide. So that is extremely exciting. And in the process, we also observed the formation of the fibrils. First, you form these donut-shaped structures from the peptide. So the peptide is lying like this in a donut. Then that collapses into small sticks. You can see a stick here, and you can see small sticks here, collapsing donut here. And then these sticks come together in long trains that mature and get you more and more solid fibers. So here is also a mechanism for how can we prevent this from happening. More importantly, this could also be a good reason for neuronal cell death, because in the neurons that are close to the Alzheimer, we see this loss of the adhesion peptide between neurons that is required for formation of synapses and communication between the neuronal cells. If they cannot do that, if they cannot form this contact, they actually die. So that could be a good reason, and here are some results from Leshinska at Allied in Nature Communications in 2015, where you see the healthy control, very little soluble protein, and in the Alzheimer, very large amount of soluble protein in different parts of the brain. Also, here you can see the healthy brain and the diseased brain, and you see a ratio of soluble to connected adhesion molecule, which is very large for some of the diseased ADEs. This is what the adhesion molecule looks like. It's connected here to the membrane of the neuronal cells, and then you have these immunoglobulin-like domains that comes together and forms the adhesion and close contact. But another thing is that these actually co-localize, so the soluble fractions co-localize with the A beta. So here you have the overlay, and you can see the beta, and the soluble fraction of the protein here is co-localization. And also our substrates in the beta is this core nucleation sequence, and it's very, very similar to the sequence that is cleaved in the NCAM, the adhesion molecule. So what we think is happening is that we form these aggregates of Alzheimer peptides that comes together and form this proteolytically active material that can cleave off the NCAM molecules, and we end up with under representation of the bound compound, the bound NCAM, so we cannot adhere the neuronal cells, and then they are, of course, dying. So we have a way to solubilize the fibrils with these two, and then we have also understanding of how the fibrils are formed and a mechanism of autoproteolytic cleavage with aggregates of Alzheimer peptides. All of these results, which could be extremely important considering that we will in 2050 have 150 million people that are affected by Alzheimer in the world. So these results are extremely important, and they are only possible because we have a daily maintenance of our infrastructure in this institute. And this is where I spend about 20% of my time just repairing instrumentation. We are back in Stockholm, and this is actually when you give your lecture. I switch them around. You're very nervous when you go up there because you're talking to 10 million people. And then you're very happy after the lecture was finished. And these are Barry Sharpless and Carolyn Bertozzi that I shared the Nobel Prize with. Now, I think that for all the reasons that I have laid out here, it's very important that we focus on education. And I think that we should make visual education within the chemistry world. And what I illustrate here is a repository where you have 5 minutes of video for every week in first grade, 10 minutes for every week in second grade, and so on. You call the AI, and the AI tells you how to get this grade this week and delivery. You get the videos, VR, text, and so on, what is required. And this is visual material, so it's very easy to translate into any language. Very easy to translate into any language, so you can have a globalization unit associated with it. I think this could be extremely important for our environment, for the climate, for global politics, and so on. Education towards excellence requires that we have good infrastructure and freedom to do what we think is relevant. So excellence is not limited to, we have had a culture of excellent people being honored. like with Nobel Prizes and other things. But excellence is not limited to the one who excels. It's actually something that happens in an infrastructure environment. So you build the excellence in a playful team, in a well-equipped playground. That means that you have the infrastructure that is necessary to do that. You also need the freedom of thoughts for all the people that are in this team, in this infrastructure. And the reason is that 5 brains are 5 times as good as 1 brain. And if you all work together, you can really get things going forward. But it's also important that it's driven by curiosity, because it's when we are really curious that we make the big discoveries. You have a true engagement because of that, and then you dream of it, because you cannot help thinking about it. And then you're allowed to do it. That means that you have the infrastructure again to be able to do what you need to do in order to investigate your chemistry. This will lead to independent co-workers, it will lead to companies and spin-outs, it will lead to a sense of self-determination under responsibility, and it will challenge the skills to move forward and the understanding of the importance of being modest. Because excellence is not something that we can create behind an office desk, it needs the infrastructure where we can practice and observe. And I'd like to give some examples, because education and infrastructure is extremely important. It's the highest return of investment anytime. Finland revised the education system, and they improved the research infrastructure dramatically, give teachers autonomy, minimal testing, equal access for everyone, and what happened is that they got fantastic performance. I am at the advisory board of the Nanoscience Center. I was at the advisory board of the Nanoscience Center in Jyväskylä in Finland, and they are really well equipped. So skills are formed in this kind of environment, and then you get economic growth, because this is like rings in the water. They just spread out into the society. South Korea and Taiwan has invested very much in education and research, and that has led to these countries being at the very front of what is going on in development and production. You had prioritized education, academic standards, and highly educated population in these countries. That led to innovation and competitive industry and economic growth. In 1999, Poland also came with an education reform, and this has led to the high level of economic growth that you see for Poland today. Vietnam focused on primary education and research facilities, and they had exchange programs with other countries that are financed from the government, and the research institutions are really well equipped. So they went from low to high income, international performance on the level, and fast economic growth. So they are second up after China and India in this growing field of economic growth. So the conclusion is that education and research lead to broad long-term economic benefits, and thus, and there should be industrial and research infrastructure is necessary. We need to develop technology at the level that is current in the world, and education investment is necessary to do that. So how can we improve the situation? Well, infrastructure hubs should be located at geolocations. That means that we would need several infrastructure hubs in Denmark, for example, associated with universities, in other countries, both with universities and research institutions. And it's really necessary to have both the instruments in the hub and also the technical people who can take care of this instrumentation and help students and researchers generate data. Because we need maintenance of infrastructure, and this is for decentralized instruments. You call for repair, maintenance is not accounted for by projects that are running because the running costs are decreasing and decreasing in our projects. So it's very difficult to maintain based on running costs. And particularly if running costs are limited, and they are today in Denmark at least. So allocated funding should be specified for institutional repair of instrument, administered by the Institute for Decentralized Instruments. An application for repair should be acquired by responsible VIPs, and instrumentation remain decentralized. I think that's necessary in the Danish system because we have a lot of instrumentation already decentralized. Otherwise, we could make an instrument hub where all the instruments would be then collectively sampled and used by all students and researchers. The instruments these instruments I show here is probably 5 to 10 million euro per hub, and an annual maintenance of 0.5 million euro, and then personnel to do everything that is necessary in order to maintain it. I think this would, research and the research cost be reduced dramatically if we could do this. And it's necessary that we do a lot because we have a need for research in the green area. It's very important for the green agenda that we find new materials instead of the plastics that are biodegradable but can perform as well as our plastics do today. We need production processes, we need new ways of making energy and store energy in particular, transition to hydrogen or hydrogen equivalents, increased food because we'll be more and more people, there's less production or more concentrated production actually. Textiles and clothing is a big problem today, transport, and there are many other areas. Most importantly, solutions to these areas doesn't come without fundamental research in molecular recognition, processing, signaling, and materials. So we should facilitate research through funding without constraints, because most important discoveries, they are serendipitous, and they are, if it was something that we already knew, it would be easy to do. So we discover something that is not expected, and that's where the novelty is. And you just have to look at the Nobel Prizes to see that that is really the fact. But we still want research to be productive, high quality, creative and novel, and we need responsibility and also translation into economy. So the way to do that today, all research money, whether it's applied or fundamental, is in one big box and we just apply for that. But I think we really have to consider that these are two fundamentally different processes. The applied research is taking the research of today, optimizing it and making applications out of it. So we have an economy. The fundamental research is for tomorrow, and that is really, really important, in particular with the rate at which this globalization and the world in general is developing. So great ideas are usually afterthoughts to odd behavior, and here are some examples. So we should have these two completely independent funding regimes and decide how much do we need for the research of tomorrow and how much do we need for transforming research today. We're back in Stockholm. This is the last day. On the first day at the museum, my wife, she saw a beautiful dress on a mannequin. And this is my wife with this dress from the mannequin. She asked the art school to actually make a dress for her for the ceremony. And you can see these are really artists that made that, because the click reaction gives you a 5-membered ring, but all these rings are 6-membered rings. So she's very beautiful, and when you dress up your wife like that, there's a real danger, and this is a danger. He is called Prince Philip of Sweden. So she was seated between Prince Philip and Prince Daniel, and I was down by the entrance in the draft. But that was okay, because the next day we were invited to the castle, and at that occasion we were 130 people, and I got the hot spot here besides coming Queen Victoria of Sweden, and you can see how goofy I look, because she's a really nice person to talk with. So thank you very much for your attention. I hope I inspired you to think a little bit different about the small-scale infrastructure instruments that are very, very important for our daily bread and butter research.
Thank you very much indeed, Morten. There are questions in the chat. So, Callum, do you want to open up some of the questions?
I'd love to. Thank you, Morten. I really enjoyed that. We'll start with a question from Susanna, who says, I really enjoyed the talk. Thanks a lot. I have a question. What were the steps in the early career that Professor Meldal has made that were crucial for the successful career, especially in relation to the access to infrastructure?
Yeah, so I think the most important thing for me was realizing how important it is to understand the structure and dynamics of molecules. And this was made feasible by my supervisor in my PhD study, because he was the one who introduced the nuclear Overhauser enhancement experiments of NMR, which can tell you what structure you have and whether you have several or a single structure, and how fast the exchange between these structures are. So I was studying carbohydrates, and I wanted to understand the interaction between carbohydrates and the antibodies in salmonella infections. And this was only feasible because we have these NMR data which show us what the molecule looked like. So for me, understanding what this NMR facility really allowed us to do, and in connection with collaborations on crystallography, we really got a full description of the structural features of the salmonella infection. And that has been sort of key to much of the research I've done subsequently, all the time doing all the difficult NMR work that is really necessary in order to understand everything. And that was the infrastructure I recommended everywhere I go. NMR and mass spectrometry is the key to the biological sciences and the chemical biology in particular. But also instrumentation that we build ourselves. I'm an engineer of education, and I love building instruments. In fact, I have built these small synthesizers here for the students in the lab. I don't know if you can see it.
We can.
I built 7 of those just recently. And that allow us to do many biomolecules that we can then study the interaction of in the bioassays that we have developed. So infrastructure, yes, it's crucial to do anything of relevance. And I think it's very dangerous for Europe not to focus on this problem in the competition with other countries, in particular China, which is coming up with the most important research at the moment.
Thanks so much.
Are leading the competition, in my opinion.
Thanks, Morten. We are on the hour now, so we just have time for one more question. I'd like to invite Miltiadis to join us on audio to share your question. We spoke a bit about this in the chat. I feel that because your question is highly detailed, you will do it justice better than I will. Miltiadis, are you happy to join us live on audio?
if he can.
Okay, perhaps not. I will endeavour to read the question out. I hope you'll all be patient with me while I attempt this. Okay. Ultiavis asks if the professor is willing to answer an off-topic question, a Venusian astrobiology question for expert opinion, as a Nobel Prize recipient. On planets like Venus, which are choked with CO2 and clouds of sulfuric acid, life wouldn't just survive the acid, it would likely use it as a resource. If you have a biological community in an acidic CO2-rich atmosphere, phosphine becomes the smoke from their fire. In an environment full of oxygen-heavy molecules like carbon dioxide and sulfuric acid, phosphine is chemically forbidden. If it's there, it means something, likely a biological metabolism, is working hard to strip oxygen away and create reduced molecules. Do you think the unknown absorbers in Venus's clouds are just mineral dust, or are we looking at the shadows of a global microbial forest? If you'd like to take a moment,
I don't need a moment. I think that Venus doesn't have conditions that promote evolution of life-like processes. In my opinion, it could be that I'm wrong. I think that the idea of life in the oceans of Europe, the moon around Jupiter, is more probable. But I also think that there are millions, if not billions, of places out there where the conditions are like they are on Earth. And when that is the case, the ability to form life is inherent in the atoms. They cannot help it. They will come together, make molecules that recognize each other, and the process will start, and it will lead to evolution of life. So that's why I think that there are probably lives, millions of places out there, but we will never be able to contact it simply because the distances are so vast that it will never succeed.
Thank you. I think there's one final question that's just come in, Callum, and maybe it would just be, because it's a nice comment, so maybe you could just, with a quick answer from Morten. Do you want to just read the one by Silka?
Yes, so Silka asks, do you have any idea how to run infrastructures in the future when the non-professorial teaching and research staff has nearly no future to get permanent contracts, for example, as in Austria and Germany?
Yeah, so that's a real problem. I think that we seriously need permanent staff, but staff that have to actually prove their value. So I think we as researchers, that's my own belief, we as researchers really have to allow ourselves to be subject to evaluation on a maybe triennial basis, evaluation that tells us how well did it go for the last 3 years. And if we haven't produced anything, there's no reason we should continue our research. If we have produced, on the other hand, very good results, the likelihood we will continue to do so is very high, and we can essentially just finance the next 3 years for such a qualified, permanent position. Permanent in the sense that if you perform, you will be permanently employed. And the other thing is that kind of system would, of course, need some entry point for young researchers. So we should still have blue sky ideas from young researchers that then they can then be allowed to prove the sanity of their proposals within a 3-year period to see if they can make any publications out of their ideas. But other than that, I don't think we should have a whole control system that tells that control the way that we are using research money. It's simply non-profitable.
Okay, thank you very much for that. And I just see that Roger wants to add Switzerland too. He feels the same pain as to Germany and Austria. So Morten, thank you very much. I think you've given us, again, some things to think about. There could be lots of other questions about the role of distributed infrastructures and maybe community, how we run things like in Europlanet across Europe, equal opportunities for people in different countries. There are just so many questions we could ask, but I thank you very much.
Yes, I could just add one comment maybe.
Please.
And that is that it's our responsibility as researchers to actually cry out to the politicians and make them change their strategy, because it's via the politicians that we have the funding for research mainly, and also in Denmark in particular from the foundations like the Novo Nordisk Foundation, the Willem Foundation, and so on. But if we don't cry out to these institutions and to the governments to finance research in a more profitable way, it will not happen. So we are the ones who know, and we are the ones that should ask for this.
I can only support you in that, and obviously you have an opportunity, you have a strong voice, and you've articulated this both at the European Summit last year, and I hope you will continue to do so. And we all have to deal with our politicians and hope that sometime they will listen to us, because otherwise the resilience in the future of European research and the next generation of scientists will be rather bleak. So thank you very much indeed. As I said, we have a slight problem is the fact that myself and Anita are at another conference, so we are going to have to leave shortly. But the other half of this meeting, or the other part of this meeting, was really just to alert people in the audience, and I can't really see who is here and isn't in the audience, but particularly for people who are in the distributed research infrastructures. So I'm just going to take the opportunity to share with you a few slides about some opportunities that are going to occur in Europe in the next few months, and put this out as a kind of a plea for people who are involved to get in contact with us if they are thinking and involved in some of these scoping exercises which are coming out. So I'm going to hope that you can see my slides. Can you see them, Callum?
Yes.
Yes, we can.
Okay, so we've talked, so in Europe, again, for some of those who may not know this, one of the main Horizon Europe programs that has various work programs, many of them are scientific-led, they are thematic areas, but across the thematic area is research infrastructures. This is how Europlanet was initially established. We are entering a very interesting period of time in 26 and 27 for research infrastructures. The new infrastructure call will open on the 10th of March with a deadline on June the 16th. Now, there are an awful lot of calls, and I haven't got time really to go through them all, but we will post these slides around. Some people on the call may know exactly what we're talking about, but others will not. But I just wanted to very quickly go through the calls that are open. And this is a kind of plea for anybody who is in their institutes, anybody who's involved in research infrastructures on this call, who are looking or expecting to participate in one of these calls, to contact us, because we would like to bring all these calls about bringing people together. And we'd like to know who's trying out things. So I'm just going to go through them very quickly. And then you can read them at your leisure. So there are 6 calls available at the moment. One of them is called Concept Development. That is the building of the next generation of research infrastructures. So if anybody is involved in the development of new research, these are fortunately a larger scale research infrastructures, not the distributed research infrastructures that Morten was talking about. But if you are involved in the development of any new infrastructures, and you're interested in trying to make the case, we would be very interested to know what sort of new infrastructures you are thinking about putting forward or upgrading. There will be, as I say, money for establishing about 4 future projects in this area. Every single call gives you a lot of detail about what you're expected to do. So you will see here that these infrastructures can be single-sited or multi-sited. This is bringing together several. So, for example, like the European Light Infrastructure, ELI, which is spread across Romania, Hungary, and the Czech Republic. So again, if anybody is working on any of these and knows of any of those activities, we're very interested to know. The next one is about bringing people together to work on how these big research infrastructures, how they can work in clusters. So in the physical sciences, you have nuclear physics, you have space, you have analytical sciences. Analytical science, obviously, being something which would be very interesting for Morten for the recent webinar. And again, these groupings are coming together. So if anybody knows of other networks, people bringing together, we know the ones in space, but if there are other ones that are being brought together to work together in building transdisciplinary collaborations, we would be interested to know. The third call is, again, looking at the longer term sustainability of these projects. Again, there's a lot of detail in these calls, so I haven't got time to go through them. But it's about looking about exactly what's just been talked about in the talk. How do we make sure that these things are going to be around in 3, 5, 6, 7 years? And again, for those of you who are obviously joining from Europlanet, you know one of the things we've done that is to form the Europlanet AISBL, an independent organization which stands alone from any 3, 4, 5 years of funding. It aims to build a structure through the community and through other opportunities so that we can operate for many years to come. One of the things that came up in those questions at the end, which is of great interest, I think, for everybody in research infrastructures, is how do we train the staff who actually operate the research infrastructures? How do we manage research infrastructures? And I think this came up in the questions very well at the end. So there is going to be one project that is funded to look at this problem. So some of the questions that were raised at the end are very topical in this. So again, if you know of people who are thinking of making a bid for this, we believe we know one group that will probably be taking the lead in this, which has looked at this before. But I think the questions that came up today, this will be an opportunity to put those forward. So how do we make sure that we train staff with the human resources to what education, curricula and courses do we put together which are related to the structure of research infrastructures? And again, I think that would be a very good, nice outcome for some of the discussions today. So we'd certainly welcome input on that. And particularly in perhaps some of the less represented countries, what's called as the widening countries. So those questions that came up at the end were very topical. Another area which is highlighted is the working with the infrastructures for working with the Ukraine. Obviously, many Ukrainian scientists can't do the research in the Ukraine for obvious reasons, and they are working outside the Ukraine. But in the future, how do we integrate those researchers into the European research area? So anybody who's got interests or is working with the Ukrainian community, we would be very interested to know what you're planning. And then there is a final international dimension. How do we link European research infrastructures with wider infrastructures around the world? Because again, as was said in the talk, research is international, and despite the global politics, scientists do collaborate around the world. So how do we bring those people together? And again, there are going to be several projects with the attempt to work about how the European research infrastructure can operate more globally. And the final call is a little bit for the technocrats. The European research infrastructure and the ERIC community, this is the structures that we have at a European level. They are working on their own strategic plan for the future. And there is a call to work there. So if any of you are involved in S3 or ERICs, we would be welcome to know. And some of those are related in the fields of climate, health, social science, etc. And then there is another call coming forward. I won't go through the details of those, just to say that this is the calls for 26, and there will then be a call in 27. And again, these will go through a similar sort of operation. So I'll stop there. We will share these slides with you. If any of those slides or any of those information gel with any of you, and any of you are working on any of those calls, we would be interested to know. I think one of them that's particularly would be of interest from Morten's talk today is how do we train these people? I mean, that came up in the question. What educational courses do you have for those people? And how can we make sure that people from different countries can share knowledge and share expertise. So, Morten, I think this might be something that you might like to might hit home to some of the things that you've raised today. So, we'd certainly like to keep you into the link as we talk to the people about that, because I think you may well have some very useful thoughts on that in the future. So these calls will be announced in 2 weeks' time. They have been around for a while, so some of you will know it. But we will be very happy to know if anybody in the call is involved in any of these things, or their institutes are involved in any of these things, so we can collaborate together. If anybody has any comments on that, or anybody would like to say that they already are involved in something, now's your time to speak. I'm anticipating that people will need a little bit of time to think. So, Callum, you'll circulate those slides after this.
Yes, of course. You can download.
And if anybody wants to talk more detail,
already.
please contact us here at Europlanet. As I say, we are liaising with others, but we'd be very interested to get people's input. And Morten, I think certainly maybe thinking a little bit about how we train people in research infrastructures and train these people. I think some of your comments on that might be very useful.
Just put this in the chat. So you can download Nigel's slides in the chat already. These can be circulated after the session as well in case you missed them. You can also contact us on infrastructure@europlanet.org. That will go to Nigel, Anita and myself. You can also contact us for all general queries on Europlanet@europlanet.org. So we invite your questions there. Okay, Nigel, is there anything else we want to cover today before we close?
No, as I said, we left it open towards the end, but as I said, I think people can look at that, and I say thank you everybody for joining, and thank you, Callum, for organising it, and most of all, Morten, thank you for excellent talk. Obviously, we have your slides, which you've kindly allowed us to circulate, and we have recorded this. So if you have other friends you want to pass it on to, please, do so. And
but can you send us all the link for the YouTube channel?
Of course.
Yes, Callum will do everything else. So, Morten, as I say, once again, thank you very much for giving up your time. And I think we can all now think about many of the things that you've said. And as I said, once again, we really are grateful for you joining us today.
Great. Thank you very much for having me.
Fantastic. Thanks, everybody. Keep an eye on the Europlanet YouTube channel for the recording of this. I'll send around a wrap-up email in a few days' time with the recording and links to all the slides so that we all have access to this information. Thanks, everybody. Have a lovely rest of your day, and we'll see you all soon.